Well, good morning, everyone. My name is Mark Optical, and on behalf of Janella's Optical Interest Group, it's my pleasure to introduce our next speaker, who probably needs no introduction. So today we're continuing our series in chemistry with 2014 Nobel laureate in the field. Of course, Eric's background is in applied physics, originally with a bachelor's from Caltech and continuing on at Cornell before going to Bell Labs. Famously, Eric left the field before coming back to academia, and together with Harold Hess, worked on photo-activated localization microscopy, for which he won the Nobel Prize. I think today Eric's going to be talking to us about his frustrations with optical microscopy, and maybe that also partially characterizes his experience with academia in the field. So we'll hear about that. Without further ado, Eric Betzig.
Okay, thank you, Mark. We've been working with advanced microscopy now for about 20 years. Both light sheet and super resolution date from the early 2000s. And they generally be considered very impactful tools. And I'm going to go through this talk and try to convince you, in my opinion, they have not been. So what are the reasons for this? Well, let's start with the fact that any image is an imperfect representation of the specimen. And I think of it as a sausage factory, that you have a biological sample that goes in, some unsavory things happen, and an image comes out. For super resolution, labeling density is always a challenge. Specificity of the label, since you're only looking at the fluorophores and not the underlying proteins you care about. Linker links then create additional confusion. And so, for example, this is a claim at the actin lamellipodium of 20 nanometer resolution. And it looks like that when you look at very sparse actin filaments further into the cell. But when you have this dense actin network at the edge, electron microscopy downsampled to 20 nanometers looks like this. Super resolution looks like that. So I've long since given up trying to fight the battle of resolution claims in microscopy because they're crazy. The other thing is because it normally takes a long time, except with SIM, to create these images. Normally people do chemical fixation to freeze the sample in place. And the EM field is known for 60 years how much damage is done by chemical fixation, by almost any means of chemical fixation. In fact, chromatin has been studied extensively, chromatin architecture by super resolution microscopy, almost always on chemically fixed cells, where the chromatin is completely disrupted, as you see by EM. And so I just throw up my hands and say, why are we doing this? You can see, even if you use live structured illumination microscopy, do fixation in place, you can watch things like the ER shrivel up and look different under the application of the fixatives. So Harold and I joined forces about 6 or 7 years ago to try to create a way of doing super resolution that would be trustworthy by doing high pressure freeze and free substitution, and then doing correlative with Harold's superlative 3D FibSem. And so we were able to do that. It's a very, very complex experiment, gives great results, but takes a long time to interpret 3D data on this scale. And also the throughput of the instrument is very low. And so there's a lot of variability in biology, and low throughput is your enemy because of that. So how general are the results of super resolution experiments? Well, again, even if you're doing live cells, the vast majority of work in live imaging or fixed imaging is by using immortalized cells on cover slips. And often these are heavily mutated lines like U2OS or HeLa or so forth, because they're exceptionally hardy. They can take the light damage that you're throwing onto them without dying immediately. But they are really strange cells. They are not typical by any stretch of the imagination. And so even if you get a result, you're left with wondering, is this valid across all cell types? Is it valid in other cell states? If you have to fix the cell, and you've already affected it by fixation, how valid is it there? And how valid is something that's on a cover slip in vivo in a whole organism? So even if you get a result that you think is biologically meaningful, it comes with many, many asterisks from what we've done so far. And again, like I mentioned before, the vast majority of results and findings by super resolution microscopy has been done with an incredibly low number of samples. And natural variability is a hallmark of life. And if you don't have high statistics for this stuff, how can you really trust what people are telling you about what you're seeing? And so much is inferred or speculated in these papers from very limited sample sets. Sometimes n equals 1. I'm as guilty of that as anybody. I'm a co-author on this paper, which has been cited 900 times now, about the organelle interactome using lattice light sheet microscopy. All of these speculations, or all this analysis, comes from one cell that was imaged, a fucking sick cell that has been labeled with 6 different fluorophores and is very unhappy. And so 900 citations, all this stuff about the organelle interactome coming from one incredibly sick cell. The other thing that you will find over and over and over again is people in the field talking about, I have this great biological result I got by microscopy. And then crickets. Where's the follow-up? They then go on to something else, and another great result, or another great result. If it's a great result, it should establish a field. It should have a bunch of follow-up papers and a bunch of work that builds upon the initial finding. That's exceptionally rare in this field. So light sheet microscopy came out about the same time as super resolution. It creates incredibly beautiful images of both cells and tissues. But there's challenges in light sheet microscopy as well. The first is that if it's applied in tissues, that aberrations and scattering will screw up your light sheet, screw up the fluorescence that comes out from that light sheet. And this is even using 2 photon going into Drosophila embryo. You can see that if you want to do lineaging in the early stages, when all the nuclei are on the surface, you're fine. And then if you show MIPS thereafter, after invagination, it still looks good. But if you actually dig in and look at the orthoslices, you see it's a mess inside. There's no way you can do lineaging with something like that. The next problem is that the aberrations vary spatially. And so if they vary spatially, you're even more screwed because you don't know exactly how to correct. And then finally, because of these limitations in doing live imaging by light sheet, the vast majority of light sheet now is by using chemically cleared and fixed specimens. So we already talked about fixation problems, but with these clearing techniques, you can get truly massive data sets, and people are getting massive data sets now. One of my biggest beefs about microscopy today is people take oodles of data, terabytes of data, and then don't do anything with it. They don't know how to do anything with it. So when I was in graduate school, I listened to a talk by Edwin Land, who had founded Polaroid Corporation, really sharp guy. And he always talked about when he's thinking about developing technologies, you got to think of it as a ladder. There's a goal up here, okay? There are many, many steps you have to climb, many, many things you have to figure out. And he always tried to find ones where there might be some missing rings in the ladder, but at least he could stretch across. Whereas other people just go to the first ladder they find, and there might be a big gap in the ladder, and they'll never be able to reach. Well, this data analysis problem in light sheet and pretty much any field now, people really flex because they can get lots of data. It doesn't mean shit if you can't analyze and get interpretable results from that data. And we really don't have the tools to do that. We can process that data. This is 500 terabytes we collected on our microscope of the mouse olfactory bulb. But trying to understand what it means and what's in it is lifetimes of work for 5 days of imaging. So what are some counterpoints to everything that I've been bitching about? And you can tell I really am a frustrated optical microscopist. Okay, so what are the counterpoints? First one is single molecule tracking. So we did this experiment with Jennifer back in 2008, photoactivating a subset of molecules. watching their diffusion to actually make diffusion maps. In this case, a protein which aggregates into virus-like particles. This one is more freely diffusing. So that shortly thereafter at Janelia, they founded the Transcription Imaging Consortium with people like James and Bob Tejan and Xavier Darzak and others to use single molecule tracking to understand the process of transcription. And again, this also pisses me off. How many times do you see, this is a beautiful looking simulation, right? And so it's like, well, how the hell did these transcription factors decide to just mosey on over to this transcription initiation complex, okay? What's happening with these nucleotides? You know, the whole thing is just a cartoon. It's Mickey Mouse, okay? It has very little to do with the reality of what's going on, all right? So the reality is that based on biochemical studies that Teej and others did over 30 years, they figured out every protein that was involved in creating this transcription initiation complex. And what happens to the biochemists and to the structural biologists and all of that is because life is live and they can't see it live, they hypothesize in their heads. They dream about what's actually going on with these proteins and so forth to have this happen. Well, they dream that you get this stable initiation complex, which then recruits the polymerase that then starts the transcription. Well, when they actually started to look with a live cell, or in this case, a live organoid, and looked at these transcription factors, they found that each one was only binding for seconds at a time. And so, like, how the hell, this picture's wrong. I mean, there has to be a different means. And so the model that started to grow is that instead there are multiple copies of each transcription factor that's needed in the vicinity of the DNA, and that stochastically they come on in a sequence and then eventually recruit the polymerase. And what causes them to all sort of cluster around that spot? They have intrinsically disordered regions on their proteins, which give them an extra stickiness that allows them to then conglomerate near that center. And in fact, it turns out that certain diseases are due to mutations in which a intrinsically disordered region from one chromosome is, by translocation, grafted onto the gene for a transcription factor on another chromosome. And then that causes an extra stickiness, which causes an overexpression of overproduction of the RNA, and therefore an overexpression of certain proteins that leads to this childhood cancer and other types of diseases. So then this led Tej and Zavi and Luke and I to say that, well, we now have a tool to be able to understand exactly how protein-proteins interactions happen in live cells and to really quantify them. And so therefore, that became the basis of us starting this company, Icon Therapeutics. So we did that in 2019. And we were fortunate enough to hire this guy who used to be the head of research at Merck. And it is just jaw-dropping. You can't see, it's behind the wall still. But it's absolutely jaw-dropping what they have discovered about protein-protein interactions in cells by single molecule tracking. They know so much more about protein and interaction networks than anybody on Earth at this point. And they're actually quite far along in the development of several compounds that may lead to actual therapeutic results that come out of this. Now, when I got the Nobel Prize and afterwards, I still wasn't sure what the fuck these single molecule techniques are really going to be good for, okay? But if this actually leads to therapeutic cures, I'll feel I've done something useful in my life at last, okay? So time will tell, but there's a lot that can still go wrong on the path to that. But it's really exciting to watch their progress. So another counterexample to this dirge of disillusionment is my lattice light sheet microscope. So by, again, having a very thin, non-diffracting sheet of light that we can sweep through single cells, we've been able to image lots of different processes in 4 dimensions. You know, I've worked on lots of tools in my life, but I think this one will probably be the most impactful once people realize what it's capable of and what it's not. And my major frustration is that this microscope has not garnered more attention by more people. People still seem to have in their head this idea that the metric that matters for a microscope is its resolution. Resolution is one metric for a microscope, but speed, non-invasiveness, all these other things are equally important for discovery. And this tool really excels at that. So what are the lessons to be learned by, after all of those things I said that I don't think we've learned much from at all, to the successes of single molecule tracking and lattice light sheet? First is that all biological structure and physiology result from dynamic processes that are occurring at spatial scales from single molecules to whole organisms. There's multiple levels of emergence leading to us. The second is that optical microscopy's unique and transformative aspect is its ability to observe these dynamics in living systems. Nothing else can do that, okay? You can't get high resolution by any other means dynamically. And so while biochemistry, molecular biology, and structural biology, to this day, still remains by far the dominant paradigms and worldviews by which biologists understand living systems, they are all basically about trying to understand a dynamic system like the operation of a car engine from a random pile of engine parts. That's what they're working with. That is fucking hard to reverse engineer, I'm sorry. And they've been at it for nearly 100 years, and they're still a long way off. So true physiological relevance requires in vivo imaging. And finally, animation and natural variability are the hallmarks of life, okay? So you have to understand that animation is what defines us, it's what defines living things. And there is a lot of natural variability, a lot of stochasticity at various levels. So the three methods, biochemistry, structural biology, molecular biology, they ignore this. And so most of biological science ignores these elephants in the room, this variability, this animation. That's what defines life. So I keep saying, how can you understand life if you don't look at it live? That is the correct final goal for optical microscopy, is to look at living systems. And in fact, optical microscopy has been used for many years to look at living systems. Furthermore, we can't just focus on looking at cells on cover slips. If I threw you naked into Antarctica, you're not going to behave the same way as you're behaving right now sitting there, right? So you need to be in the environment in which you evolved. And so people have been doing that with 2-photon and other tools that you can see here. But again, it gives you a little bit better than single cell resolution, but doesn't really give you much information about what's happening on the subcellular scale. So because of the lattice light sheet, we wanted to be able to do in vivo imaging. It was also defeated by the aberrations. So we developed an adaptive optic approach using a 2-photon guide star to determine the aberrations, and then deformable mirrors and a spatial light modulator to correct the aberrations for the light sheet going in and the fluorescence coming out. And so this is showing how much gain there is in zebrafish by actually applying adaptive optics, in this case 2-photon, this case confocal. So that then led to, I don't know why this movie isn't playing, or all these movies aren't playing, but this led to our adaptive optical lattice light sheet microscope we published in 2018 that has those 2 AO arms on our lattice light sheet. This is looking at spinal cord development in zebrafish. This was going to be a neutrophil moving in the inner ear. This is showing 3 stages of cancer cell extravasation from the rolling motion with long, sticky filopodia that come out. That's even better. Cancer cell that they eventually attach to the blood vessel and crawl, and then they extravasate. And then this is looking at development in the eye. So, all right, well, I'm losing it all, so I'm going to have to bounce out of the program for a minute and go back in and find out why we're doing this. This is slide 19. Let me try this again. Sorry about that. Okay. There we go. All right. So The problem with that microscope was a 10-foot train wreck on an optical table that cost $1 million, and nobody else would want to build something like that. So we started to work on the development of a second-generation instrument of that would be more compact, cheaper. Once you realize all of the components, like a 2-photon laser, linear lasers, galvos, detectors, mirrors, blah, 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 you realize that you have all the components for pretty much every microscope that's been designed since 1990. So why not actually make it a Swiss Army knife that can do all of those modes, all with adaptive optics? So that's what we call this thing, mosaic. There's 2 of them here at Genelia. There's 2 at our Advanced Imaging Center. There's been about 40 research licenses to date, but I'll get into what that all means later on. So here's some of the modes of the mosaic. There's an upright station where we can do AO 2-photon imaging. So this is going in the somatosensory cortex, showing with and without AO the improvement. We can do oblique illumination to kind of see the whole cellular outlines. We can also do functional imaging with the 2-photon. We can do 3D single molecule localization over fairly wide fields of view. We can use lattice light sheet over very large fields of view, either as a regular lattice light sheet or in a mode where we do one-dimensional structured illumination in the direction of the light sheet to get higher resolution, as you can see here. We can actually apply that then in fish to actually do AO lattice sim inside of zebrafish, as you can see in this example. It has, like I said, a very big field of view, and basically its speed is only limited by the cameras. I mean, most good light sheets now are simply limited by camera speed in terms of acquisition rates. So this is basically at 4 terabytes per hour, 100 terabyte data set, looking at a field of dividing LLC PK1 cells. Now, the next challenge, of course, is when you're generating 4 terabytes per hour, is you can't store the resulting 100 terabyte data sets over and over again before your drives are completely full. So you need essentially real-time pre-processing of that data and compression. So Zhang Tao and Matt developed in our Advanced Imaging Center at Berkeley the means to take light sheet data at that speed, compress it, convert either to TIFF or ZAR formats, and then do first a D-skew rotation of the raw data, then stitch different tiles together with different adaptive optic corrections in each tile to deal with the isoplanic patch problem into a single volume, and then deconvolve that. And that can all be done now on about 24 cores. You can do right at this 4 terabytes per hour. So we can do real, and then we compress that final result. So we now have, this took a long time, but now we have a solution that really matches the speed of the microscope to do all of that. But that's just that first part. We stressed that system by, again, doing the mouse olfactory bulb, where we work with Ray Gao, who's an expansion expert. We did 2x expansion of the bulb. Because of the limited working distance of our objectives, in order to be able to see the whole thing, Ray developed this photocleavable gel. So it's sort of somewhat similar to like FibSim, where you basically image a certain slab, then you cut off that slab with UV light, then you image the next slab, and on in order to get the whole thing. So again, then we can look at much bigger samples with that. This is then showing the overall view of that bulb. Now, that's fine, but all that is still, it's now processed data, but it's still bits on a disk, which isn't useful. And furthermore, if you're taking 4 terabytes per hour, you don't want to go many hours blind and not knowing whether your data that's streaming out is worth anything. So you need some kind of real-time visualization in order to do that. We've been working with NVIDIA, who has developed this index platform, a multi-GPU solution that allows you to do real-time manipulation of the data. If my movie would play again, you could see that. So again, just in real time, as you get the data, you can then manipulate the data. You can do slices through the data. You can follow time courses, zoom in, and so forth. And so that's still a bit of a work in progress, but it's developing. And all my movies are starting to slow down. So, so that's fine. So now we have the ability to take data at high rates. We have the ability to pre-process data at high rates. But what we don't have is the ability to then analyze the data at high rates. Oops. The challenge with multicellular imaging and the promise of multicellular imaging is that there's, of course, in biology, many different cell types, many different behaviors. The advantage of the lattice AO is that we can see all of those. So this is an experiment where we cut the tail of a developing zebrafish, and then this represents the response to that wound, cell debris, cells moving into the wound area, the epithelial cells on the surface anchored to the other cells with this dynamics of fibrils to the underlying layer. In the mesenchymal space, you get different types of events like fusion of cells. Once the cut is done, the capillaries actually grow and remodel over time in order to get more stuff to the cut. And then neutrophils are flowing in towards the cut region as well. So lots of different types of events are happening inside it. This is life. You have 37 trillion cells in your body doing this kind of dance right now. Okay, so this to me is the future of biology, is being able to understand that. So how do we quantify and understand that? Well, in the two papers we did in 2018 and 2019 with our AO lattice light sheet and then the expansion lattice light sheet with Ed Boyden's group, each of these papers generated quite a bit of data, and GoCool spent about a year on each of these papers and about 3 million CPU hours on each paper on the Janelia cluster in order to do sort of custom coding to analyze what was coming out of that. Well, this is not exactly a scalable solution because as much as I'd like to, I can't clone GoCool many times over. So we have to find other ways to be able to analyze data at the terabyte to petabyte scale. So this is where my proposal lies, is in trying to, I'm the biggest skeptic about ML in microscopy that you will ever find. And yet I have been led to believe that ML is probably the only way we're going to get analysis tools that are scalable at this size of the data. So you can see that the data coming out of the microscope is great, but it's way too complex for the human eye to be able to interpret. So we need some means. So of course, one approach is to use transformer architecture, because by knowing the relationship between the sequence of the words was key to being able to get a large language model that succeeded by using, again, that transformer architecture that maintains that understanding of the spatio-temporal relationships of the data. Well, there's spatial-temporal relationships by the zillions in this data. And so we think that this is the right approach to take. This is the transformer architecture. It was used first by Google in order to create a means of classifying 2D images, scraping off 45 terabytes off the web, 300 million images, pre-trained in 140 days on 100 equivalent H100 GPU hours. Meta then took the Google VIT and then fine-tuned it for the task of segmentation inside of images in this beautiful example that you can see here. Well, when you have data like I just showed you from the lattice light sheet, the first challenge is segmentation, right? So having a good AI segmentation tool would be good. So extending this now from 2D plus color to 4D plus color is what we would have to do. Not necessarily trivial. This took, had to have 6 million annotated images, took 430 days just for this 2D version here. But if you could do that, in this case, this is what GoCool did, using Elastic to basically, again, analytically segment the cells. But if we could be able to use a transformer model to do that, we would be first a huge step ahead. Because if you want to understand subcellular dynamics in multicellular organisms, the first task is being able to tell where a cell begins and where a cell ends, so you can understand where these boundaries are and what's happening in each cell. So how would we do that? How would we be able to train for segmentation? Well, nature's already done that. Nature and Jeff Lichtman, I guess, and his crew, by doing Brainbow, right? So we can do stochastic multicolor labeling to get a biologically annotated data set of segmentation, and then be able to use that with a membrane label in fish, ultimately then to be able to train to be able to segment cells when they just have the label, the membrane label. So once you get to the point of then segmenting out a cell, then you have to start look at the subcellular dynamics and try to extract insights from that. So this is again GoCool doing an analytical custom coding example to look at how the organelles change as this cell is going over time inside of a tissue. This is not an isolated cell. This is a cell in a developing fish in the brain, and seeing how these organelles change. You'll also note that, for example, like the Golgi doesn't look anything like the Golgi that you normally see in a cultured cell on a cover slip. And that tells you, again, the limitations of looking on a cover slip, because things are different in different cell types and in different developmental stages. So that means that we not only just want to segment, then we have to do the analysis on those segmented cells. And there's lots of tasks to try to first understand what the cell type is, what is the state of each cell, classify the organelles, filaments, condensates inside of each cell, then characterize the interactions between these components, how they remodel themselves over time, and then if we can also throw into an extended foundation model, a multitask foundation model, we can then throw in the biochemical information and genetic information as well to see how they correlate to these behaviors, or pharmacological perturbation as well. And then finally, in the science fiction-y version, what you would have is many, many, many petabytes of multicellular data with subcellular resolution that would then be addressable to biologists through a large language model. And they could just say, please tell me what happens to the mitochondria in this type of organism when a neutrophil has to go through a confined space. And that's how you would interact with it. It would start to give you data information related to that. Hell, it could fucking write the paper for you and you can go and retire, I guess, or maybe we'll all be put out of work. I don't know. But this would be the dream, okay? Many, many years away. I won't be alive to see this probably, but I think this is the path forward. So what are the obstacles to this? Well, first is you need the microscopes in order to do this. And people have to be incentivized to follow this particular path. So we've tried 3 different approaches to getting access for people for microscopes. The first is open access, like our AIC here at Janelia. So this has been a very popular facility to be able to use, like our lattice light sheet, Harold's FibSem, and other techniques. But it requires a lot of resources. You can see that it really relies heavily on the resources that are only available at places like Janelia. It's hard to appreciate how much these things matter until you try to do it yourself over in Berkeley like we've done. Berkeley is not Janelia. Berkeley doesn't have the resource of Janelia. Very frustrating, and every time I come here I feel very jealous at the resources that you people command compared to the shit we have at Berkeley, okay? So at Berkeley we have 2 of these mosaics. I could tell you stories that would curl your hair, but anyway. And we're trying to do it on as much work as we can. So a lot of other places have tried to copy the AIC model, which is fine. But even so, in nearly a decade of operation, despite all of this, there's a major problem, which is that 2 major problems. One is that you can't push that many people through those few instruments. So there's not that many projects you can take on. And the second is that a biologist, a 2-week window of experimentation is usually not enough. It's around and then they say, oh, fuck, we still have to do this, this, and this. They have to come back again, but no, that's another funding round of proposal. So it's 6 months to a year before they're back. This is not tenable, really, and it's not really easy to get high stuff. So in nearly a decade, the lattice, which has been the dominant performer, 30 papers, 3 a year. On this level of resources, so many people, such a budget, such an incredible place this is, roughly 3 papers a year, 1.5 papers a year, 1.6 papers a year. So, and the other thing is that another thing that has always pissed me off, and I've worked with dozens and dozens of collaborators with my microscopes, particularly the lattice light sheet, is that oftentimes the imaging is just meant to be the cherry on the top of some biochemistry or genetics paper so they can push it into a better journal, okay? It's not central to the point of the paper. It's just there to try to get it. You're talking to the #1 cynic on earth, folks, so I'm sorry, but that's the way I view it. So you can't push many people through that way. Well, the mantra of the advanced microscopy development people for the last 20 years has been open source, do everything we do open source. Well, we spent an incredible amount of time, maybe 50% of my group's time for several years, to open source each of our microscopes. With this microscope, the lattice light sheet, over in axon dendrite, we put cameras around the room like an operating theater, took it apart, put it back together again, image, 16 hours of video you can download with hundreds of pages of documentation. Every little bit. Dan there has done the lion's share of the work of all of this documentation, and we have a Google group where you can go in and Dan answers their questions and so forth of that. So roughly over 130 research licenses have been given out on the basis of this microscope. Probably, I would venture to say, of that, maybe 40 scopes were built. I'd guess maybe 15 are still in use. So that's pretty pathetic for the amount of effort that went into it. But why is that? Now we get to the incentives part, okay? The guy who wants that microscope is a biologist who has a question or a series of questions. But usually he's not an engineer, okay? So that means that all of a sudden he has to become an interdisciplinary, vertically integrated group that hires engineers and physicists. First, he's got to find the right kind of guys, which is tough. He feels uncertain because there's this loss of control. There's shit happening in his group he doesn't really understand, okay? The second thing is he usually hires students and postdocs who are still wet behind the ears and don't know one end of a screwdriver from another when they start. Next, there's misaligned incentives because these postdocs or graduates and say, well, hell, this Betzig group already made this thing. I'm not going to get a career by making a copy of this thing. You know, I have to do something else. So they do some kind of tweak on it or something, publish some paper, kind of goes into some shitty journal and then goes nowhere in the end. And so that happens over and over again. And finally, even when they're good, they fucking leave. They get their degrees and then they're gone and then you're starting all over again with somebody else. And so it sits and it collects dust. It's not a tenable model for broad access, okay? This open source model, it isn't. People are living fairy dreams by believing that it is. Okay, hang on. So the final thing is, well, also, how did I come to this slide? I don't even remember how I was coming to that. Oh, yeah, the other problem for use in other people's groups is you still have to analyze the data. In my talks after the lattice light sheet became popular, I had a laugh line I would use, which is saying that everybody goes away with 10 terabytes of data and big smiles on their faces because they've seen the systems they've studied for 20 years in ways they've never seen before. And then they call us up a month later crying because they have no idea what to do with 10 terabytes of data. And that's still true. It's still true. They get great data. They can see when we render movies, they go, oh my God, I can't believe this. And then they don't know what to do with the data. And so, and it's got, with Mosaic, it's worse than ever. You know, 800K, 4 terabytes per hour. Well, even for us to be able to do that real-time pipeline that we have, we had to build our own $2.4 million cluster and have the storage and have the compute to be able to do all of this stuff. And all the visualization workstations and all the rest, 3 times as much as the damn microscope costs. Not many groups have not many academic groups have the resources to do this kind of thing. Not even many imaging cores have the resources to be able to do that. And then for us, we had to be vertically integrated in this because we couldn't afford even to go on the cloud. We needed to do a solution that for us would do what the cloud would do, but would do it locally. But the problem is all this stuff depreciates really fucking fast. There's a new generation of GPUs every 6 months. there's better hard drives every year. So we're burdened. This is a problem. And the final of the three paths to access is commercialization. And I will say that things like the AIC, ABC, and the instruments that we have successfully gotten into other labs to work have had a beneficial effect because they create a demand. And so it gets you over the valley of death so that some commercial outfit will say, yeah, maybe it actually is worth trying to make a commercial version of this instrument. And it succeeded in that regard, and now there's 3i sells a lattice light sheet, Zeiss sells a lattice light sheet. My optical lattice stuff is in the Zeiss Sim product. But despite all of that, I can't give numbers because they're confidential, but let's just say that the sales of all of these instruments has been underwhelming, to say the least, much less than we thought when we did the licensing. Part of it is because it's not marketed properly in the case. So Zeiss, to my bewilderment, almost always shows 2D maximum intensity projections of lattice light sheet data when they start to look like fucking confocal images then. And they're static, and like, this is not what this instrument is for. They sell it on the basis of its high speed and low toxicity and not on its 3D imaging characteristics. I think in part because they feel people are scared of the 3D imaging, and they rightly say, how am I going to interpret that data, right? And so they do it on 1 or 2 occasions, show a 3D rendering. So why have these microscopes? And I think this is true of nearly all super resolution microscopes, commercial super resolution microscopes. Even when they sell, they're not used. I mean, every time I go out and give a talk in the outside world, if I'm in academia, I ask the people, what instruments do you use in your lab? Can I see your lab? And if there's an imaging core, I want to talk to the imaging core people. I want to find out, what instruments do you have? What instruments are used? Well, 80% of the time, it's this bad boy, okay? It's been around for 400 years. That's what biologists use. Why? Large fields of view, simple and robust for a biologist. Even if they smash the damn objective into the slide, it's not a big deal. Inexpensive, lots of contrast mechanisms, fast and non-invasive. Undefeated champ for 400 years. About 80% is that. About 10 to 15%, well, let me just say that it's so popular that predictions are that it's still going to be used 300 years from now. Okay, so the other one is the confocal microscope, because the wide field's major limitation is you can't really look in thick tissues with it. So you need something that has optical sectioning capability. Well, you would say that STED and Palm and all these techniques, SIM, can do better than confocal, but confocal has been a damn moving target, okay? So like, for example, Zeiss's Aries scan, which uses this 32-element array, can basically do ISM-type resolution enhancement to nearly SIM-type resolution. And so if you show a biologist, I can use this crazy wide field microscope that can only look at thin samples because of out-of-focus light, but is non-invasive, and I can get microtubules that look like that. I can use a STED microscope, which is pretty damn invasive and pretty complex to align and keep aligned in my lab for long term. And I get an image like that. I can do single molecule localization microscopy and spend months figuring out how to label this thing at high enough density and high enough specificity to see the thing I want. Or I can get that Aries scan right off the shelf, and I can immediately get images that look like this. Now, there is not really, for most biologists, if they're not diving right down into the nanoscale, there really isn't any difference to them in terms of its utility of this versus this, because this is nearly SIM already. It's pretty fucking good. And it's faster because now they have a sensitive detector with multiple elements. They're not filtering through a tiny pinhole anymore. So it's got improved resolution, improved sensitivity, improved speed, reduced invasiveness. The detectors can run from the UV to the IR. It can do optical sectioning that this can't do and this can't do. And this has trouble doing if there's any aberrations. It also can be applied to FRET, FRAP, FLIM, FCS, 2-photon, all these modalities on one scope. So if you're a biologist saying, do I want to get this lattice light sheet? Do I want to get this STED? Do I want to get this area scan? It's a pretty fucking easy decision 9 times out of 10, okay? So we don't move the needle, okay? So the final thing is, if we want to do what my proposal is to be able to find the trouble with things like confocal and wide fields, they'll never be able to do what I want to do, which is to be able to understand subcellular biology in multicellular organisms. Confocal is too slow, too damaging still, particularly as you go in depth. You never get away from the fact that you have a lot of out-of-focus fluorescence that's being generated and has to be tossed away. So lattice light sheet with adaptive optics, I think, is the ideal tool to approach this problem. But the problem is the size and complexity of the data. So I already talked about using a foundation model to be able to do this. How big and complex would this foundation model would be? That is really hard to tell, okay, because it's hard to know how to extrapolate from what's been done. But again, starting with the Google VIT as a start, they looked at 18,000 classes of images, over 300 million images, and training time was 3,400 hours on an H100 equivalent. So if we want to do pre-training for the model that we're talking about for zebrafish, and we say that we're looking for either 50 to 500 different types of classes, which could either be cell types or organelles in the cells or other things, then you're talking about anywhere from 0.8 to 8 million images. These images are actually movies, okay? So that many movies. The data set size is anywhere from 0.4 petabytes to 4 petabytes. That doesn't bother us at all. We do need to create a high throughput microscope to do that, but that's the easy part for us to generate that data on that scale. Training time, though, starts to look pretty ugly. So anywhere from, this was stressing Google several years ago, less of a stress now, but you're talking anywhere from 30,000, depending on how ambitious you want to get in terms of the number of classes, up to 300,000 H100 hours. And the number of fish that you would need to create lots of fish, to create lots of diversity, to cover lots of cases and lots of different types of organelles. So you're talking a pretty damn big pipeline in order to get this done at scale. But then that's just the start. That's the pre-training. Then you have to apply to, fine-tune it for different tasks. Let's look at segmentation. If you use Meta again as an example, they took 6 million annotated images of this size. The training time was 10,000 hours. If we try to extend that to the zebrafish, we're talking this many cells, this size, and you end up coming at around 15,000 to 50,000 hours to train. So again, doable, particularly with Brainbow as a way of getting that annotated data. I think that's plausible. But still, all of this is really, really, really pushing both the architectures of the foundation models and the amount of compute that's needed in order to do it. We do have Perlmutter, a supercomputer up on the hill at Berkeley. If we could sell this to them, maybe we would get access. It would certainly have more than enough horsepower to do what we want to do if we can get that data up there to there. But anyway, at this point, it's asked. The other thing is, these numbers are just a linear extrapolation. Who the fuck knows what's linear and nonlinear in terms of the scaling? It could be completely off, okay? In terms of, there's sort of like a quadratic dependence in these foundation models in terms of looking at associations and patches of images, near term and long term. So linear scaling may not even be plausible. So it's aspirational, okay? So putting the pieces together, what will we need to do this? It requires expensive and complex microscopes, a high throughput lattice light sheet, hundreds of transgenic specimens, supercomputer level compute and storage, and even hardest to come by world-class ML expertise when these guys are across the bay making 5 times what they could ever work for us, maybe in some cases 10 to 20 times what they could get. So you have to get the nuts who want to do something cool as opposed to the ones who care about money. There's not a lot, but there's a few. Whether we could attract enough, I don't know. So because of all of these things, I am a guy who has always wanted to work at the bench himself. My group has never been more than about 3 people at any one time. I think that's why we've been successful, and I can rant about that later if you'd like, about small groups versus large groups. But this is not a small group endeavor, I'm afraid. And so in my opinion, this is something that actually requires pretty much its own institute in order to do this. It requires an independent, vertically integrated research center with aligned incentives. Is there a model similar to this? And there is, and that's astronomy. Okay, so astronomers don't peer through their own telescopes at all. What they do is they come up with a proposal for something that they want to study. They send that proposal to the observatory. The proposal is accepted. The observatory itself does the imaging, and the observatory itself does a lot of the analysis as well. Sometimes the astronomers do it themselves, but a lot is done by the observatory itself. So I think we're headed towards that type of model to address things on this scale. So therefore, I propose a cell observatory with the mission to explore the complex and dynamic space within the cells of living organisms. The inputs would be proposals from biologists as well as orthogonal data. They may have biochemical, genetic, et cetera, structural, that could be put into the foundation model as well to correlate. The outputs would be first an open source specimen. If the whole thing falls flat on its face, at the very least, we will have a vast database of subcellular cell biology inside of living tissues where all the cues that lead to the correct expression of proteins' behaviors occur, which does not happen on a cover slip. So that would be valuable in itself. Also, open source processed image data. The specific investigator would be given an ML analysis of the specific questions he posed. And like I said, ultimately the dream would be that everybody would access post-publication with the foundation model and be able to interact with it through an LLM to ask biological questions from that. So that would be the dream. So whether I will ever find somebody willing to fund that dream, I don't know. I'm just at the beginning of that process. I have a feeling it's going to take quite a while if I succeed at all. So the last thing I'd like to say is there's been so much talk over how super resolution has been a revolution for biology and how light sheet is revolutionary. Well, when is a revolution a revolution? Let's look at quantum mechanics. From 1900 to 1930, you went from no knowledge, you went from Planck figuring out the radiation is quantized to Dirac coming up with the relativistic Schrodinger equation. And most of that work was in 20 years. In nuclear energy, Hahn and Meissner split the nucleus in 38. In 42, you had the first reactor with Fermi, first bomb in 44, first fusion bomb 52, first submarine in 55, first commercial reactor in 57. 19 years, folks. 19 years. 20 years of super resolution and light sheet. What are the big hits? Okay. What is our revolution wrought? What have we figured out? Okay. How were we better off? I leave that question for you. Okay. And I'll end there. Thank you. Any questions? Disputes? Arguments? Yeah. Oh, well, we need a, yeah, there, the box. There you go.
Very, very intriguing this, but I would like to, what's your opinion about the clarity of samples? I mean, we have a limitation of what kind of biological samples we can
Yes.
look at.
Yes.
Most of biology
Is
doesn't
not
really look
clear.
at transparent
That's right.
samples.
That's right. Absolutely.
So-
we have to do what we can do, right? So we have to use transparent organisms to begin with, model organisms. In my opinion, this is a step forward over looking at cells on cover slips, and because you have the right environmental cues, but I agree you're only looking at a small subset. The reason people look at zebrafish is at least it's a vertebrate. So you're at least a little higher up the evolutionary chain. But yes, there will still be plenty of dangers about trying to extrapolate what you learn about metastasis in a fish from what you learn about metastasis in a human. No question about it. I'm fully on board. I wish I had a way of making a cleared human being. It would be kind of gross, but kind of fun at the same time. but sadly, I don't have that. Yeah. Next. Oh, come on. I was really hoping for... Is anybody at EMBL ready to argue with me?
I'm happy to argue, but not.
Okay.
But actually, I have a question before that because I really liked your
Yeah.
spot-on analysis and especially this idea about creating, let's say, a cell observatory, right? But then, like in physics, the question is a little bit like who will judge and evaluate those proposals? Who will basically decide
Yeah.
what's the biggest challenge out there to tackle with something like this, given all the
Yeah.
funding that would need to be put into this, right? Like who judges
Yeah,
the grand challenge?
I think you'd have to attack this very much in the same way that Geneli itself was attacked when it started, right? Is that, Jerry had a number of workshops to figure out what were the most interesting questions and things to push on. So I think having a lot of external input on that regard would be helpful. So TBD, I guess, is what I would say. Yeah.
Yeah, I guess one can learn a lot from the physics community, as you've pointed out, with the particle accelerators and the like. It was very similar,
Yes,
right? I mean, people have to agree early on to actually have a chance to convince.
Although I think there has to be, you have to find, I guess, what's the word, a board of directors or something for long-term stability of the right mission. Another problem that I have, and another reason I've always disliked large organizations, is I think they have limited lifetimes before they kind of senesce, I would say. I would argue that things like the ALS at Berkeley or some of the stuff they're doing, I was just at Oak Ridge, they're not going to like me for saying this, but they have currently today the world's fastest supercomputer. And when I look at the problems they're using it on, I go, why? A lot of molecular simulations and stuff we can just do it bigger and at finer time resolution than we did before, but there isn't any real. So there really has to be some people who are over in charge to keep things focused. I feel Genelia has really lost its way over time in terms of as it's gotten bigger and other incentives have come in play versus just scientific excellence. And the idea originally very small groups, which I still firmly believe in. Again, that's another topic. But I like to have input, but I think Jerry's eventually having sort of a dictator in the end, kind of making ultimate calls or, supported by some Sidney Brenners and things like that, is a pretty effective model. It was a model that worked for the LMB under Perutz. It was a model that worked at Bell Labs for 50 years. It worked here for about 10 years. So that's the kind of model I'd like to see. Yeah.
Thank you.
Great. I think we have another question.
Yeah. Yes. On EMBL. Yes. Your hand's up there. Yeah.
Yeah. Hi. Thanks. This was a really interesting talk and indeed provocative. But I go back to what was in the abstract, and in the beginning you talked of how reductionist a lot of biology is.
Yeah. Here's the abstract. I just put it on the screen there for you.
And but that we're not looking at these things live, right?
Yes.
So, but you never addressed how we would do biochemistry, look at genetics and biochemistry in the molecular level live. And you did mention for your big model that you would throw all of this data into your model.
Yeah, you want to find the correlations between what you've learned by biochemistry and so forth to the behaviors that you see dynamically, right? That would be the hope, to find those correlations. Yeah.
Yeah. Okay, but you're quite provocative in suggesting we throw out a lot of the reductionist approach.
I'm not saying we throw it out. You misunderstand me. I'm not saying let's kick the biochemists to the curb. Far from it. What I'm saying is that they've ruled the roost for 100 years. They still rule the roost. I'm at Berkeley. I'm in the MCB department. I know how many people do these techniques, and they still dominate. Whereas what the imaging could potentially accomplish by doing live subcellular, multicellular imaging is certainly not appreciated. And even the tools like lattice light sheet and what it can tell them about their biochemistry and their molecular biology and structural biology. Just as, again, SPT taught the biochemists a hell of a lot about transcription that they had wrong, right? there's still so many opportunities for live imaging to be able to tell those people that these models they've created in their head, instead of their Western blots is the truth.
Yeah.
Their models is speculation, right?
Yeah, no,
So,
I totally agree. Like I'm on the microscopy live imaging side.
Yeah.
I just wanted to clarify that.
Yeah, sure. Yeah. Okay, cool. Okay, another one on the left there.
Yeah, Peter Park, please.
Yeah. The
No,
gold shirt. Yeah.
Yeah, hi. Okay,
Hi.
you can hear me, right? Cool.
Yes.
Yeah, I really love to hear this idea of like, because we have just like too much data, the scale across temporally and spatially, it's just too much to process, and that's why we have this need for like a foundation model that we can use deep learning to extract some kind of biological insight. But I noticed that most of the task that you're considering, it's mostly just like segmentation of
That's just the initial, I guess, ante into it, right? You have to segment the cells to know where cells begin and end, right? To know whether if I want to study something about, mitochondrial morphology, I better know whether this is in this cell or this cell and what that cell type is, right? And what the cell state is and other things like that before I start making some conclusions about morphology of mitos, right?
Yeah, and
so it's just a start. Yeah.
I think it's the right way to get a start on this. My question is, I would love to hear your opinion on like, given this like so much data, like as a kind of a data-driven approach, like maybe what do you think about like the way to maybe enhance the image based on all this data that we have? Because using deep learning, you can also maybe interpolate between pixels, or interpolate between temporal frames.
ML image enhancement, what has been gained and at what cost? That's other talking points I have, right? So I still remain a skeptic. Most of the papers that I see using DL for image enhancement, for example, to sharpen up a sim image or like that, I've looked at a lot of cells by super resolution. I know what they look like. And I know that a lot of times when I see DL applied to things, you'll see microtubules that vary in intensity. Fuck, they don't do that, okay? There's so many monomers per unit length and that's it, okay? So if I see a microtubule that looks, a lot of the DL applied to these images ends up looking, I call it baked. They look like roadkill a little bit. They have this sort of like, super high contrast, super sort of like it's been, like it's been like a worm baked on a sidewalk they stepped on kind of feel, right? That doesn't read true to a lot of, for example, the sim imaging I've done, okay? I'm not saying that it can't do it, but I'm saying that and a major problem of that field, and I think a major challenge and threat to what I propose is hallucinations. And I think there has to be a huge emphasis on coming up with ways of doing quality control to make sure that things aren't hallucinating and how to deal with it when it does. And I don't know exactly how to do that. I think that's a long discussion to figure out, sort of standards for the field to figure out how to handle hallucination.
Hey,
thanks.
Jeff Rhodes. I worked in CellMap doing the
there you
machine
go.
learning
There you go.
segmentation, and I worked on image enhancement for high energy X-rays. In grad school.
Yeah.
So I think this is one.
I'm
delightful
boring your
talk.
rocks then.
Huh? Oh no, this is perfect. I agree with everything you said.
Okay.
So publication in grad school was about how we need to look at a lot of the architectures that have been developed in the private sector, specifically CycleGAN, which they bake in these incentives to make the networks work better, easier, faster, because their domain is generally non-critical, you know, in segmenting a natural image so that
Right.
you can pick out yourself on your
Right.
photo on your phone
Right.
is cool.
Yeah.
We're not trying to get like a truth.
Right.
Not only that, the huge like bazillion priors.
Right.
2D image.
Exactly.
Heads tend to be at the top.
Yeah.
Stuff like this.
Yeah.
And so I'd say, yeah, there is a lot of work to still do. I think a lot of these, like the image enhancement thing that CellMap's doing, is trying to lay the groundwork to build up and be able to figure out what can we take from the private sector. You know, how do we adapt the vision transformers so that it can actually be used on huge data
Right.
sets.
Right.
And then also, if we want to make a good foundation model, we don't want to just have one modality.
Absolutely.
Right?
That's not what a
So-
foundation model is about. Right?
Exactly. So
It's
having
about being
like
multimodal.
starting
right?
to make these massive
Yes.
repositories of segmented data.
Yes.
It's going to take time, but I'm very optimistic. And I
Yeah.
think that you've laid
I
out
just think that you should
exactly-
apply your expertise to the live data instead of the EM data. That's what I would argue.
one step at a time, right?
Hi, my name is Cedric. I'm physicist, but I'm doing machine learning. I would disagree with the large language model because
what model?
The large language model.
Yes.
The
Yes.
foundation.
Yeah.
I'm not sure this is really needed. If you point the revolution that you have pointed, like the
Yes.
quantum physics, and this is only one revolution that you pointed. There are 3 in total.
Mm-hmm.
There was no large language model, so maybe we don't need this large language.
No, that's true. But I would say in response, I've worked with a lot of biologists. The bar for what they can actually pull into their own brains is pretty low, okay? And so if they can talk to somebody and get some kind of information that way, that is way, way easier for them than to try to, if they have to code anything, or if they have to go on a screen, or they have to even look through a database and click, you know, like the databases that Janelia puts out for CellMap. Most biologists completely, your neuro whatever, glancers, they don't know what to do with that shit. They, you know, they're somehow we have to meet them on their own terms because they're the customer, okay?
My point so is that I think it's a language question.
Yes.
So to follow with the research on the language, I think it's worth even so for physics, even so for all. So to understand the language, to propose a new language to describe what we described, I think it's worth. But the foundation model as a must, I'm not sure. But
I view the LLM and the foundation model as basically the interface. I don't view it as foundational to the foundation model. And I think people with a more advanced mindset can interface with the foundation model without going through the LLM, right? But the biologist needs something they can understand.
Right, but if you make a large foundation model that recapitulates everything, you did not improve anything because you have to understand the foundation model. So it's just a replicate of the biology with another stuff that will not be understandable.
I didn't quite understand that.
Yeah, we have to.
Okay, we can talk offline.
thank you.
Yeah, all right. Anything else?
Eric, a quick question.
Yo.
Prediction. Do you think there's going to be more companies in the future that does what your company is doing, or how's that going?
What ICON is doing? Good question. I don't know. I do think that you know, I keep telling Roger, I'm on the SAB for ICON, and I keep saying, you know, sooner or later you're going to want to move away from cultured cells and actually look at this stuff in organoids or other things, right? And so there will be, obviously, at some point, a natural progression of that, in which case I hope to get more involved in that. I think other companies, you know, I know, you know, like Alex, what's his last name? The guy who did the T-cell stuff with us? Yeah, Alex Ritter. So he ended up at Genentech using a lattice light sheet there, right? So there's a few like seed things into pharma in order to do this sort of thing. I think Zeiss has more of an aspiration to sell things like their lattice to pharma than they do to academia, because academics are a pain in the ass to work with generally, and pharma is either both more experienced and more tolerant of not everything being perfect. But yeah, I don't know ultimately how to do this properly. My feeling is that I didn't have enough time. I talked a little bit about it in regard to the problems with open sourcing lattice light sheet and how they fall into disuse and so forth. that you know, the incentives in academia are not incentives to get to science. The incentives are to publish papers and get grants, okay? And so everything works towards those incentives. And so I think ultimately the real science, just like ICON is, like I say, ICON and their knowledge of intracellular protein interaction, they know more than anybody else on Earth, no question about it. And why? Because the incentives are aligned. You've got biologists and chemists and ML people and imaging people all working towards profitability, IPO, big paycheck, stock options, blah, 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 blah. That's a powerful incentive. I don't know if it flashed on the screen or not, but Charlie Munger, who was, until he died, you know, co-head of Berkshire Hathaway, always said, show me the incentives and I'll show you the outcome. Okay, super true. And in academia, the incentives usually are not about scientific discovery. They're about career enhancement and expanding the base of the money that comes in through the government and through tuitions in order to keep expanding that. And so, and then it creates large organizations, large groups that are inefficient because of something called Price's Law, which is another topic for another day. Yes, yeah.
So on the...
you can see I love being at Berkeley, by the way. Yes. Okay.
On the transient student postdoc problem, part of this vision. So given cell observatory, maybe this is just being trained as a biologist. I have a failure of imagination on like how physics students are trained, how astronomy students are trained. How do you reconcile the idea of like transient training with, like, does this fit into this cell observatory model? Are they proposing projects that
I would prefer to hire people who are beyond that level of inexperience, who like to think of this as a career, right? Or ones that you can, the SpaceX model, okay? So you get people who are postgraduate, who have some experience. You run them through 10 years until they're nervous wrecks and they leave, and then you get and it refreshes the organization. The thing that Janelia lost was when they didn't refresh the organization often enough. The thing about Bell that was great is that, you know, every year they rank people 1 to N, like the NFL draft, and the bottom 10% was told to find the door, right? That provides a powerful incentives to keep going. If you start to feel comfortable, and Janelia's been all about, I've watched Janelia's comfort level rise over the time, and it just results in mediocrity, and complacency. So, anything more? Should I leave it on that happy note?
I'll try to keep going.
Yeah.
I mean, one of the other things about open source is, you know, you shared all this information, these schematics with everyone else, but I'm not sure if it really went the other way, right? The open source software, the pleasures that everyone else also did contributes back to your project. And one thing I've noticed about, say, Google and Meta is, you know, they came up with a way to get everyone else to share their images so then they could build these foundation models.
Yes.
And so I'm wondering, how do we do that trick with biologists? How do we get them to share all their images with us in high resolution?
Yeah. Well, first off, they don't take it at high resolution. Often, I would not trust the imaging data I get from most biologists today. Now, that's a little bit different than like scraping information from various sources off the web. You know, I mean, one Taylor Swift image is as good as another, right? But it's a little different with the kind of data we want. I do believe that ultimately the foundation model would have to extend to other organisms, other problems, which would come from outside imaging data. But that data would have to follow a certain set of protocols and procedures for quality control before it starts polluting the foundation model. Anything else? Yeah.
So should people give up on open source essentially?
It's up to you. Okay. I cannot tell you how much time we put in open source. I would wager we put in more effort in open source than anybody. The Mosaic too, right? I mean, what's there, 1,000 pages of documentation on how to build a Mosaic? Yeah, about, yeah. And again, there's probably about 40 people who've done a research license. How many would you say are even operating in lattice light sheet? Like 7, 8, 9, something like that? How many are operating in lattice AO? 4? The 2 at Janelian, the 2 here? How many are operating in all the modes? 1, 2. The 2 at the ABC. That's it. Most people are happy just to have a good lattice light sheet microscope, if they do it, but most of them don't get that far. It was a mistake making the mosaic. The thought at the time was, okay, well, if we have to spend all this money to make an AO lattice, and it can do all those other things, well, I've been to enough imaging cores where you walk in the door and 50, 60% of the microscopes are dark, and they have to yet, because the imaging core head has to somehow predict what their user base is going to need in terms of imaging, they have to cover all of their bases with all of these techniques. And the thought was, well, if we could have one microscope that would rule them all, then they could press a button and they can service all of those different people with one machine. It would have 100% utilization. They could have a few copies of that and be done. Okay, that was the thought. But even for us, and we are the best, I will say without embarrassment, we are the best at building high-end microscopes. It took us forever to get a mosaic that would be aligned simultaneously for all those modes at once, so you can push that button and be a confocal or a 2-photon or a light sheet. We did it, but that took 3 or 4 more years than I thought it would take. So it was, for what we want to do for this foundation model, I would like to build a very simple inverted AO lattice light sheet, similar to like the Bruker one, where it has a little FEP thing, and you have 2 objectives that come in like that, and you can have your zebrafish all lined up in a row and go bzzz down there and do that. The hallmark of anything that you want to do on an industrial scale is robustness. It's the other thing that's amazing about ICON is they have 30 plus swept high-low light sheets running 24/7. They collect a petabyte of data a day. It goes directly into MLs and out. They don't do localizations. It directly pops out all the histograms of diffusion histograms, K on, K off, all of this stuff, all at that petabyte per day rates with 30-odd microscopes running balls to the wall. So it is possible to do these technologies in an industrialized kind of format like that. I think the same is true for the lattice AO if we get that going. Cool. All right. I guess I'll leave it at that. Thank you, everybody.
Thank you.